we shall do certain important questions on the animal organization lesson here now. I shall read out the question for you. For the first question is, the earliest recorded attempt classification of plants and animals was done by A. Linnaeus, B. Whittaker, C. Aristotle, D. Bohin. Now, among these, Aristotle made an attempt to classify the organisms for the first time. So, Aristotle should be the correct answer. And about this, you have already learnt here now different types of classifications here now. Two kingdom classification was put forth by Linnaeus, where Linnaeus divided the organisms into two kingdoms, Animalia, kingdom Animalia and kingdom Vegetabilia. That is, he placed all the plants under Vegetabilia and all animals under Animalia. Then later on, the three kingdom classification came into existence. Three kingdom classification was put forth by Ernst Haeckel. He divided the organisms into three groups here now: Protista or Protoctista, then Plantae, then Animalia. That he placed all the unicellular organisms under Protista. All unicellular organisms under Protista. Then with the discovery of the bacteria, scientists found importance of Monera also as a separate kingdom here now. So, two kingdom classification was put forth by Linnaeus. Three kingdom classification was put forth by Haeckel, where he divided the organisms into Protista or Protoctista, Plantae and Animalia. Then next, four kingdom classification was put forth by Herbert Copeland. Divided the organisms into four kingdoms, that is, there was an addition of Monera, Protista, Plantae and Animalia. Okay, Monera, that is, included all prokaryotic organisms under Monera, all unicellular organisms under Protista, then all multicellular autotrophic organisms under Plantae, then animals, that is, all heterotroph organisms under Animalia. That is, there was no separate place for the fungi in the four kingdom classification. So, in order to create a separate kingdom for the fungi here now, five kingdom classification came into existence. Five kingdom classification was put forth by R.H. Whittaker. Divided all organisms into five kingdoms here now. The first one is Monera, kingdom Monera. It is the kingdom of unicellular prokaryotes. The next is Protista. It is the kingdom of unicellular eukaryotes. The next one is Plantae. It is a kingdom of multicellular autotrophic organisms. The next, Animalia. It is a kingdom of multicellular heterotrophs. Then next is fungi. It is a kingdom of multicellular saprophytes. Then the five kingdom classification was put forth by R.H. Whittaker. So, in the question here now, he says earliest recorded attempt of classification, it was done by Aristotle. So, the first question, correct answer. So, the first question correct answer is C, Aristotle. The next question number 2, bacteria that live in most harsh habitats are A. Eubacteria, B. Cyanobacteria, C. Mycoplasma, D. Archaebacteria. Archaebacteria are the bacteria which live even in harsh habitats like extreme cold, extreme heat, sulfur springs, okay, so on and so, even salt pans, so on and so forth here now. So, Archaebacteria is the correct answer for second question. So, question number 2, the correct answer is D, Archaebacteria. Archaebacteria can live even in salt pans also. We call them Halobacteria. Then the bacteria that live at high temperature, thermophilic bacteria. Okay, the bacteria which live even in cold areas, isn't it? Like different habitats they live, hostile environmental conditions. The bacteria which can thrive well are Archaebacteria. So the second question correct answer is D, Archaebacteria. The next question number 3, two kingdom classification was given by, just now I made a mention here now, Whitaker A, B Linnaeus, C Copeland, D Haeckel. So two kingdom classification was put forth by Linnaeus. Third question answer is B, Linnaeus divided the organisms into two kingdoms, kingdom Animalia and kingdom Vegetabilia. So third question correct answer is B. Then the fourth question here now, fourth question, five kingdom classification. Five kingdom system of classification is based on A, mode of nutrition, B, complexity of the body organization, C, complexity of the cell structure, D, all. Whitaker, while classifying the organisms, he took all the characters into consideration, that is on the basis of mode of nutrition, on the basis of their complexity of the body, whether they are unicellular or tissue graded or organ graded or organ system graded. Then on the basis of complexity of the cell structure, that is very prokaryotic or eukaryotic. So on the basis of all these characteristics, he divided the organisms into five kingdoms. So fourth question correct answer is D. Now question number 5, blue-green algae cyanobacteria are A, heterotrophic protistans, B, parasitic animals, C, photosynthetic monirans, D, saprophytic fungus. Now these bacteria, they are all placed under monera here now. Once again the bacteria, you have Eukaryote, then blue-green algae, that is cyanobacteria, then Archaebacteria. There are three types of bacteria. All of them are placed under monera. So in question number 5, he says blue-green algae are, so they are obviously due to presence of chlorophyll, they are photosynthetic monirans. So fifth question correct answer is C. Now question number 6 here now. In Whitaker's five kingdom classification, eukaryotes were assigned to A, all five kingdoms, B, two of the kingdoms, C, four of the kingdoms, D, any one of the five kingdoms. Now eukaryotes, the first one is monera, it includes prokaryotes. The rest of the four kingdoms are eukaryotes only, that is protista, eukaryotic unicellular organisms, animalia, multicellular eukaryotic heterotrophs, plantae, multicellular autotrophic organisms, fungi, multicellular heterotrophic saprophytes. So the rest of the four kingdoms include eukaryotes. So sixth question correct answer is C, four of the five kingdoms. So sixth question correct answer is C. Now question number 7 here now, two kingdoms that are common to all biological classifications. A, monera plantae, B, plantae animalia, C, protista monera, D, animalia and fungi. When you look into all the four kingdoms, that is two kingdom, three kingdom, four kingdom and five kingdom, the common among them are animalia and plantae. So seventh question correct answer is B. Now question number 8 here now, monera includes A, unicellular eukaryotes, B, saprophytic eukaryotes, C, prokaryotes, D, all of these. So as I made a mention earlier, monera includes exclusively the prokaryotic organisms like Eukaryote, cyanobacteria or blue-green algae, Archaebacteria, PPLO, pleuropneumonia like organism, Rickettsia. So all of them are referred to as prokaryotes. So eighth question correct answer is C. Kingdom of eukaryotes with chitinous wall. Kingdom of eukaryotes with chitinous wall. A, monera, B, protista, C, fungi, D, cyanobacteria. Now among these here now, in monera bacteria, cell wall is deposited with muramic acid, peptidoglycan, so on and so forth here now. And protista, the cell wall may be deposited with cellulose, or sometimes it is deposited with silicia, that is silica, so on and so forth here now. Cyanobacteria, once again the bacteria, peptidoglycan, murin are the compositions of cyanobacteria cell wall. So C, fungi, in fungi here now the cell wall is chitinous, deposited with chitin. So the ninth question correct answer is C. Question number 10, kingdom monera includes A, bacteria, B, cyanobacteria, C. Archaebacteria, D. All. All of them belong to Monera. As I made a mention earlier, Monera includes prokaryotes, which are Eubacteria, Cyanobacteria, Archaebacteria. So the answer for the 10th question is D, all the above. Now question number 11, cocci bacteria in cubical form of 8 or multiple of 8 are known as Diplococcus, B. Streptococcus, C. Sarcina, D. Streptobacillus. The cocci bacteria, that is rod-shaped bacteria, are known as cocci bacteria. When they are arranged in cubical form of 8 or multiples of 8, they are known as Sarcina. So 11th question correct answer is C. 12 you have. The term bacteria was coined by A. Leeuwenhoek, B. Ehrenberg, C. Louis Pasteur, D. Robert Hooke. Okay, the term bacteria was coined by Ehrenberg. Okay, bacteria were discovered, studied in detail by Leeuwenhoek. That's why he is also called father of bacteriology. But the term bacteria was coined by Ehrenberg. So 12th question correct answer is B. 13th question, which of the following is not a characteristic feature of halophils? A. Presence of mucilage sheet, B. High potassium ion concentration, C. Special lipids in the cell wall membrane, D. Enzymes function at low pH. Here halophilic bacteria, there are two types, halophilic bacteria and halophobic bacteria. The bacteria which can thrive well even in high salt concentration are called halophils. The bacteria which cannot live in high salt concentration are called halophobes. So among these characteristics, functional cell wall, enzymes functional at low pH. Okay, so that is not the characteristic feature of halophilic bacteria. Okay, so 13th question correct answer is D. Enzymes functional at low pH. It is not a characteristic feature of halophilic bacteria. Halophilic bacteria, when they leave a high salt concentration, obviously alkalinity is very high, so enzymes are not functional at low pH. So for 13th question correct answer is D. Enzymes functional at low pH. Now 14th question here, it is about once again Archaebacteria. Which of the following is thermoacidophilic Archaebacterium? A. Thermoplasma, B. Halobacterium, C. Methanobacterium, D. Methanococcus. Thermoplasma are the bacteria which can live even at high temperature. Halobacteria live in high salt concentration. Methanococcus bacteria are the bacteria which live in the gut of ruminants. Methanococcus bacteria are the bacteria which are capable of producing methane. They also live in the gut of ruminants. So he says thermoacidophilic, that is the bacteria which can live at high temperature. So obviously it is thermoplasma. 14th question correct answer is A. Thermoplasma. 15th question here now, read out for you. The nitrogen fixing bacteria in the roots of legumes is A. Halobacterium, B. Rhizobium, C. Thermoplasma, D. Methanococcus. Among these, A, C, D, they are all Archaebacteria. B is the true bacterium that lives in the root nodules of Leguminaceae family and thereby leads symbiotic life. So nitrogen fixing bacteria in the roots of legumes is B. 15th question correct answer is B. Rhizobium. Now 16th question here now. The cell wall material of most of the monerans is A. Peptidoglycan, B. Pseudomurin, C. Branched chain of lipids, D. Cellulose. Now peptidoglycan is the basic component of the cell wall of the bacteria. So peptidoglycan, okay, 5-amino salicylic acid, peptidoglycan, murin are the ingredients of the cell wall of the bacteria. So among these options, the correct answer is peptidoglycan. So 16th question correct answer is A. Now question number 17, four kingdom classification was proposed by A. Aristotle, B. Leeuwenhoek, C. Haeckel, D. Copeland. I made a mention earlier, four kingdom classification was put forth by Herbert Copeland. He divided the organisms into Monera, Protista, Plantae and Animalia. So 17th question correct answer is D. Copeland. Now 18th question, I read out the question for you. The sugars present in pseudomurin are A. N-acetyl glucosamine, B. N-acetyl isomurominic acid, C galactose, D 1 and 2. So the pseudomurin, which is there in the bacterial cell wall, is contains both acetyl glucosamine and acetyl isomuronic acid. So 18th question correct answer is D. Now question number 19 here now. Match the following. We have list 1 and list 2. List 1 is about scientists, list 2 is about discoveries here now. A. Hugo Debris, B. Frankel Contract, C. Hatch-Slack, D. Buchner. On the list 2 you have 1. Enzymes, 2. C4 pathway, 3. Genetic nature of RNA, 4. Mutations. Then options are A. A4 B3 C2 D1, B. A4 B2 C3 D1, C. A3 B2 C1 D4, D. A1 B2 C3 and D4. Now we shall try to find the correct answer here now. First, if you take A. Hugo Debris. Hugo Debris. You know very well that Hugo Debris is the father of mutations. So has put forth the concept of mutation. That is, fourth option is the correct for A. Hugo Debris. Now next, next B. Hugo Debris, Frankel Contract. Frankel Contract, B. He is the one who put forth genetic nature of RNA. Put forth genetic nature of RNA. So the third one, Frankel. Correct answer is third one. The genetic nature of RNA was put forth by Frankel Contract. Next, Hatch-Slack. Hatch-Slack, you know very well, is the one who put forth, both of them put forth C4 pathway. So for the C, Hatch-Slack, what is that C4 pathway. So the correct answer is 2. Next, D option, Buchner. Buchner is the one who studied about the enzymes, coined the term enzyme, also studied about the enzymes. So first option is correct. So A4, B3, C2 and D1 is the correct option. So among these, correct option is A4, B3, C2 and D1. So 19th question correct answer is A. 19th question correct answer is A. Next, question number 20. Triploblastic animals exhibit A. Asymmetry, B. Radial symmetry, C. Bilateral symmetry, D. Spherical symmetry. Most of the triploblastic animals barring a few exceptions, most of them exhibit bilateral symmetry, wherein the animal is divisible into identical halves by a median sagittal axis, that cuts animal mid-dorsal to mid-ventral direction. So 20th question correct answer is C. Now question number 21. All the members of Animalia are A. Eumetazoans, B. Unicellular, C. Multicellular, D. With tissues. Now all members of Animalia are, most of them once again barring exceptions like maybe Chlamydomonas, Euglena and all that, most of them are multicellular here. So all the members of Animalia are that is, if you we take as classification here now, Animalia includes the animals right from Porifera onwards. Porifera to Chordata, all of them are placed under Animalia. Protozoa, they are unicellular, they are placed under Protista here. So according to Viteka, Protozoa, which is unicellular, they are also animals but they are placed under Protista. So as per R.H. Viteka's classification, all the members of animals are multicellular. So 21st question correct answer is C. Question number 22. Cellular level of organization is exhibited by A. Sponges, B. Coelenterate, C. Protozoans, D. Cnidarians. Now sponges or poriferans are the animals that exhibit cellular grade of organization here now, where the cells are loosely organized here now, do not form tissues due to lack of sense organs and nerve cells. So 22nd question correct answer is A. Sponges exhibit cellular grade of organization. Now 23rd question here you have tissue level of organization is shown by A. Sponges, B. Coelenterates, C. Nematoda, D. Porifera. Now tissue grade of organization is noticed in Coelenterata or Cnidaria and Ctenophora. So among these options, the correct answer is B, Coelenterata. So 23 question, correct answer is B, Coelenterates or Cnidarians are the animals that exhibit tissue grade of organization, and diploblastic animals with outer ectoderm or epidermis, inner endoderm or gastrodermis. So 23rd question, correct answer is B. 24th question here now. Organ system level of organization is exhibited by most A, sponges, B, coelenterates, C, diploblastic animals, D, triploblastic animals. Organ system grade of organization is noticed right from Platyhelminthes onwards. So among these options here now, D is the correct answer because all organ system grade animals are triploblastic. So 24th question, correct answer is D, triploblastic animals. Now 25th question, alimentary canal has a single opening in the animals that belong to, okay. Alimentary canal has a single opening, that is sac-like gut it is referred to as. It is noticed in A, Coelenterata, B, Platyhelminthes, C, Nematoda, D, Annelida. So in coelenterates there is no alimentary canal at all, they have got only gastrovascular cavity. Platyhelminthes are first formed enterozoans, but however the gut is incomplete without anus, so single opening is present in Platyhelminthes, that is sac-like gut is present in Platyhelminthes. Nematoda, first time well-developed gut is noticed with both mouth and anus, and Annelida onwards the digestive glands and well-developed gut is noticed here. So 25th question, correct answer is B, Platyhelminthes. Now 26th question here now, Siloam is formed by splitting of mesodermal bands in A, Platyhelminthes, B, Archaebacteria, C, Echinodermata, D, Annelida. If the coelom is formed by splitting of mesodermal blocks or mesenterblasts, it is called schizocoelom. We already learnt about this. The true coelom is of two types, schizocoelom and enterocoelom. The coelom that is formed by splitting of mesoderm is called schizocoelom. In schizocoelomates, the mesoblasts or mesentoblasts, which make their appearance at the junction of outer ectoderm and inner endoderm, they give rise to mesoblastic bands. Each mesoblastic band segregates to form mesodermal somites, and later on each mesodermal somite splits at the center to form outer somatic or parietal layer, inner visceral or splanchnic layer. So among these options the correct answer is D, Annelida, because Platyhelminthes acoelom is present, Archaebacteria pseudocoelom is present, Echinodermata it is enterocoelom. So D, Annelida is the correct option with schizocoelom. So 26th question, correct answer is D. Now question number 27, type of symmetry with only one central axis but many planes of symmetry is A, bilateral symmetry, B, radial symmetry, C, asymmetry, D, biradial symmetry. Now symmetry in which only one central axis but many planes of symmetry. In radially symmetrical animals, all the body parts are arranged along the oroeboral axis, that is anterior to posterior, so that the animals can be cut into identical halves by many planes passing along oroeboral axis. That's why it is also called multipolar, heteropolar, multi-axial symmetry. It is also referred to as heteropolar multi-axial symmetry. Heteropolar it is called because both mouth and anus, mouth is at the oral end and the other end is called basal disc or pedal disc without any anus and the body parts are arranged oroeboral axis. That's why it can be cut into identical halves by many planes along oroeboral axis. So it is heteropolar monoaxial. It is heteropolar. Heteropolar it is called because both oral and aboral end are entirely different here now. Monoaxial you call it here now because along one axis it can be cut into identical halves by many planes. So it is heteropolar monoaxial, that is radial symmetry. So 27th question, correct answer is B, radial symmetry. 28th question here, read out the question. Body forms left and right antimeres in only one plane in A, sea anemones, B, amoeba, C, hydra, D, ferritima. Body forms left and right antimeres in only one plane in. When it is cut here now, you get only two planes, that is right and left halves. So the correct answer is D. Ferritima, because in bilateral symmetrical animals, animal is cut into two identical halves by a single median axis like this. It can be cut here now from dorsal to aboral so that you get two pieces. One is towards right, the other one is towards left here now. So 28th question correct answer is D. Ferritima is the correct answer, that is bilateral symmetry. So 28th question correct answer is D. Next, 29th question here now is the leader of the question for you. Which group differs from rest of the three regarding symmetry of adults? A. Echinoderms, B. Annelids, C. Coelenterate, D. Ctenophores. Which group differs from rest of the three regarding symmetry of adults? Now, echinoderms, if you take here now, adults are pentamerous radially symmetrical while larvae are bilaterally symmetrical. Okay, then annelids are bilaterally symmetrical. Then coelenterates, if you take, they are radially symmetrical. Ctenophores, they are radially symmetrical or biradially symmetrical. So which group differs from rest of the three regarding symmetry of adults? Okay, you can say Echinodermata adults are radially symmetrical, Coelenterata radially symmetrical, Ctenophora also is radial or biradial. So among these, only B is the odd one out, because in Annelida adults are bilaterally symmetrical. So 29th question correct answer is B. Annelida. Now question number 30. Siloam is lined by A. Ectoderm, B. Peritoneum, C. Endoderm, D. both endoderm and mesoderm. You know very well here now, Siloam is a cavity that lies between outer somatic or parietal layer, inner visceral or splanchnic layer. These two layers are also referred to as peritoneal layers. So 30th question correct answer is B. Peritoneum. We have question number 31. A body cavity is not lined by mesoderm occurs in A. Aschelminthes, B. Annelida, C. Platyhelminthes, D. Echinodermata. Now in pseudocoelomates now, body cavity is present but it is not lined by mesoderm here now. Because if you take Annelida, it is true coelom, Shagocoelom is present, that is not the answer. Echinodermata, true coelom, that is entocoelom, that is not the answer. Platyhelminthes, there is no coelom at all because body cavity is deposited with parenchyme or mesenchyme. So Aschelminth is the correct answer because pseudocoelomates where the body cavity is not lined by peritoneal layer or mesodermal layer. So 31st question correct answer is A. Aschelminthes. Now question number 32. Platyhelminthes are characterized by A. Radial symmetry, B. Diploblastic nature, C. Acoelomate nature, D. Complete gut. Platyhelminthes are acoelomates where the body cavity is obliterated by the mesodermal tissue called parenchyma or mesenchyme. So platyhelminthes are characterized by acoelomate nature. So 32 question correct answer is C. Acoelomate nature. Now question number 33. In a garden, presence of earthworm can be traced by the presence of A. less fertile soil, B. more birds, C. worm castings, D. less burrows. Now you identify the presence of earthworms in the garden by means of worm castings. Fecal pellets of earthworms are called worm castings. So you find piles of worm castings here now. It is an indication that there are plenty of earthworms in that particular region. So 33 question correct answer is C. worm castings. 34th question here now. Dorsal side of earthworm is marked by A. nerve cord, B. genital pores, C. dorsal blood vessel, D. genital papillae. Now earthworm here now you can identify dorsal and ventral surfaces by two characters here now. Dorsal surface is dark, ventral surface is pale. Dorsal surface is dark due to presence of a pigment called porphyrin. Apart from that, on dorsal side a dark line is present along the mid dorsal side here now. That is due to the presence of dorsal blood vessel just below the skin. The dorsal blood vessel is the thickest and the longest blood vessel in earthworm. It appears externally as a dark line. So by means of the dark line, that is dorsal blood vessel, and porphyrin pigmentation, you can distinguish between dorsal and ventral surfaces in earthworm. So 34th question correct answer is C. dorsal blood vessel. 35th question here now. In earthworm, mouth is present at the anterior end of A. peristomium, B. cingulum, C. pygidium, D. clitellum. Now the earthworm here now, mouth is in the first segment here now, that lies in peristomium, which is also called first segment or the buccal segment. Peristomium is overlapped by a fall segment called prostomium. It is referred to as false segment because it does not contain any viscera and does not originate from pygidium. So mouth lies in the peristomium and it is overhanged by prostomium. So the correct answer for 35th question is peristomium. A is the correct answer.